Hello, everyone, and welcome to today's live broadcast, CRISPR and ZFN Mediated Gene Editing for the Exploration of Neural Cell Mechanisms and Modeling, presented by Jeremy Liam, Product Specialist Functional Genomics, MilliporeSigma. I'm Alexis Corrales of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by MilliporeSigma. For more information about our sponsor, please visit www.emdmillipore.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box that appears on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Jeremy Liam. I will now turn the presentation over to him.
Thanks, Alexis. My name is Jeremy Lehmann. I've been with MilliporeSigma now for almost 10 years. Most of that time is spent in research and development, working with ZFNs to perform genome editing experiments, and now most recently working as a CRISPR Product Specialist, doing technical consultation, guide RNA design, donor design, among some other things here in my current role. MilliporeSigma is the life science business of Merck KGaA out of Darmstadt, Germany, operating here in the U.S. under the name MilliporeSigma. So we have a long history of expertise in gene editing. We've been in the exclusive supplier of zinc finger nucleases for research purposes for the last decade. And we're one of the first companies to commercialize CRISPR reagents. We've received multiple awards for our innovations and offerings in the CRISPR space. In 2015, with an award for the CRISPR epigenetic activator based on dead Cas9 fused with the P300 histone acetyltransferase domain. More recently, in 2016, an R&D Top 100 award winner for the Sanger arrayed lentiviral CRISPR libraries. Now in 2017 and 2018, we're coming out with new libraries for CRISPR-based activation with the SAM library. So let's jump right into it. When people talk about using CRISPR or CRISPR-mediated gene editing, we're usually talking about nuclease-based creation of double-stranded breaks in DNA, and these are targeted to a specific sequence in that DNA. When using these technologies, ZFNs or CRISPRs, we're relying on endogenous DNA repair mechanisms to achieve the desired modification. The outcomes are achieved through either non-homologous end joining that you see on the left or homology-dependent repair that you see on the right. There are some tools and strategies that can shift this balance a little bit, but it's not really your choice which mechanism the cell uses to repair the double-stranded break. And it most often uses non-homologous end joining, which is error-prone and creates a mutation at the site of the double-stranded break, often termed an indel, which is a word for a combination of the words insertion and deletion. So the full extent of CRISPR's utility really extends beyond targeted cutting of DNA, though. You can use it as nuclease-independent applications where you're using CRISPR as a tool as a cargo delivery method. So this is a closer look at how ZFNs and CRISPRs work. They both create targeted double-stranded breaks in DNA, but there are key differences between the two technologies. ZFNs are composed of a DNA binding domain made up of protein that is fused to another protein, the FOC1 nuclease. Each zinc finger recognizes 3 base pairs and is linked to either 3, 4, or 5 other zinc fingers to bind up to 25 different bases of DNA specifically. And these 2 zinc fingers come together to cut DNA when in close proximity. As you can imagine, though, generating custom DNA binding proteins for every sequence you want to target can be quite laborious. This is partly why CRISPR technology has been so revolutionary. The CRISPR system uses just 20 base pairs of complementary RNA sequence to target DNA before binding with the trace RNA and recruiting the separate protein Cas9, the nuclease, which comes in and cuts DNA. One of the key requirements for Cas9 is the PAM site, which you see there in blue. PAM sites are what makes any given sequence of DNA eligible for the creation of cRNA and cleavage by Cas9. A PAM site is a sequence of DNA, a guanine dinucleotide, or NGG. The tracer RNA exists to tie the cRNA, the targeting molecule, and Cas9 together, and it is an invariable sequence of RNA that can either be connected to the targeting portion of the RNA or left separately for delivery in ribonucleoprotein applications. So CRISPR-based genome editing and ZFN genome editing as a tool can be used to achieve a wide array of genetic modifications. Gene knockout is perhaps the most simplest application shown here, and we'll go into more detail on targeting these first 2 features as well. Things like transcriptional activation are possible, as well as fusing a reporter gene or making specific sequence changes like you might see in a disease modeling of a single nucleotide polymorphism mutation. But first, we want to take a moment to talk about how we might go about fusing a reporter gene. What you see here is an example of some work we've done internally almost 10 years ago now, where we tagged the actin gene with a red fluorescent protein. To do this, we designed ZFNs targeting very close by the start codon for this gene and integrated a red fluorescent protein just at the N-terminus. We used homology arms that were 848 bases and 593 bases in length, the left and right side respectively, and again integrated this red fluorescent protein as a fusion to the coding sequence for the actin gene. This allows us to visualize that cytoskeletal element and create some really nice images via fluorescence microscopy. But you can do more than tag just one gene at a time, though. You can also tag multiple different elements within a cell. In this particular cell line, we tagged the nuclear cytoskeletal element, lamin, with blue fluorescent protein, and as well as tubulin with GFP. This creates a triply labeled cell with all these different cytoskeletal elements visualized via fluorescence microscopy. These are actually reporters of the cytoskeletal elements in question, not overexpression systems as well. So really interesting for any experiment that you're trying to do. Next, I want to take and go over a similar application where we're fusing fluorescent protein and creating a reporter gene or reporter construct. This is a publication that was done in 2011 using zinc fingers to tag a specific gene that is expressed in the development of neural tissue. This neural tissue is named the adenohypophysis, which is really another term for the anterior pituitary. And the reason that these researchers want to tag that tissue so that they can study the development of that tissue. The tissue is responsible for the production of glucocorticoid hormones, things like thyroid stimulating hormone, ACTH, as well as LSH. So let's take a closer look at what they did in this specific application back in 2011 with ZFNs. But first, take a look at the gene expression network that's at play and during development of this particular tissue type. They differentiated embryonic stem cells into oral ectoderm, which is characterized by the expression of the PITX1 gene and PITX2 gene before they get to the gene that they tagged in this experiment, which is responsible for the formation of these Rathke's pouch progenitors in the anterior pituitary tissue, LEM3. LEM3 expresses a luteinizing hormone and follicle stimulating hormone, like talked about earlier, among some other glucocorticoids. So it's a very important tissue to be able to model and study if you're looking at neural development processes. This is an illustration of the donor construct that they use to integrate the fluorophore behind the LEM3 gene. When we're talking about a donor construct, we're talking about a template for homology-directed repair. In this application, they used 1 kilobase homology arms on the left and right side, equally sized. We see that these homology arms can be different in length. They don't have to be exactly the same. But if they are the same, that seems to be an easy way to move forward with the design of your donor construct. Again, much like the cytoskeletal reporter line I showed you earlier, they targeted the N-terminus of this gene to integrate their fluorophore, so creating a fusion just behind the start codon. And something they did different in this experiment was include a mode of selection on their integration cassette. That mode of selection is antibiotic resistance cassette, which is flanked by LOXP elements so that they could excise that particular sequence of DNA, leaving behind only the reporter gene fused, which is LEM3 fused to the venous fluorescent protein. So let's take a look at some of the data that they were able to generate. And the cultures that they created to differentiate into LEM3 expressing tissues are called large cell aggregates, which are cultured under the conditions containing a sonic hedgehog agonist molecule. So in order to induce expression of LEM3. And you see this nice increase in expression in fluorescence, which is directly driven off of that reporter construct that they made. The figure I here is a flow cytometry analysis. Figure J, an image of these large cell aggregates at a low resolution. K, you start to see even higher resolution here. Some of the pouch formations, and that's extracellular formation of these Rathke's pouch components. And then when you take a cross-section of those large cell aggregates, you start to look even closer, you can see the LEM3 specific cells there in figure L. These are the cells that are responsible, for the expression of and secretion of those glucocorticoid hormones. So tagging a reporter gene really isn't the only thing you can do with CRISPR or ZFNs. Of course, you can do more simple things like gene knockout, but those can also be used in more complicated experiments. So here we move on from fusing a reporter gene into a gene knockout application. This is a paper that was published using some of our reagents in 2016, targeting the PCM1 gene. The PCM1 gene is implicated in several different oncology studies as a marker that might be useful for targeting as chemotherapeutic agents. PCM1 depletion inhibits the growth of ciliogenesis, and it increases cell death and sensitivity to temozolomide. When we take a look at any gene that we want to study for gene knockout, it's important to understand the structure within there and the architecture of that gene. The human PCM1 gene is a rather large and complicated gene. It has approximately 9 kilobases of coding sequence, 51 different available transcripts, 39 different exons. And when you process the coding sequence for this gene, you get 44 different available 3 base pair mismatch guide RNAs. So it makes it difficult to choose which guide RNA that you would want to move forward with for gene knockout. Some of the rules that we like to employ when we're doing these gene knockout guide RNA designs are that we target within the first 2/3 of coding sequence and outside of the first 90 base pairs. We target outside of the first 90 base pairs to assure that we don't run into any issues with alternative start sites. Sometimes in the very beginning of coding sequence, you can have an alternative start site and then pick up gene expression after you've induced a mutation in coding sequence. But again, as early on in the coding sequence as possible. Then you also want to select a guide RNA that targets, that is common to all different transcripts. So coding sequence among these different transcripts is often shared. And we want to target coding sequence that's common to all 51. And you see here that some of the data for the guide RNA that was chosen for this experiment, that it targets all 51 different transcripts. It has a high specificity score as well as a acceptable efficiency score. We see specificity scores are pretty accurate. Efficiency scores less so. We see typically guide RNAs with scores above the 30 mark for efficiency have good activity in cells, but there's not a lot of correlation between guide RNA activity and the efficiency score besides that. So take a closer look at some of the data here in this publication. The L0 and S3 are two different glioblastoma cell lines. Within those cell lines, two different clones were made, wild type clones that were just purified and gone under the same genome editing processes, but with non-targeting constructs, and then PCM1 containing clones or PCM1 depletion clones that you see nearby. And we measure the percent of ciliation, which is implicated in metastasis of these cell types. We see the clone F9 and clone B9 here with a significant decrease. We also see that these PCM1 knockout clones are more susceptible to the chemotherapeutic agent temozolomide or TMZ, which is what you see in the figure on the right. So this experiment was done with plasmid DNA. They actually sorted these glioblastoma cells based on GFP expression of a plasmid containing Cas9, GFP, the guide RNA and the guide RNA for gene knockout. But there are several other applications with RNA and even lentivirus. Lentivirus is a growing area of CRISPR-mediated gene editing. So let's take a look at another publication that was some of our products using lentivirus in the application into an animal model. This study looked at the reversal of hypofrontality in animal models of addiction and schizophrenia. Hypofrontality is common in cell models of addiction and animal models of schizophrenia. It's characterized by a reduction in activity in the prefrontal cortex. So let's jump right into this paper and take a closer look at some of the data that they were able to generate with gene knockout using lentivirus. Wanted to briefly state the genome-wide association studies that they had indicated SNPs in the human chRNA5 gene. And that's the gene that we're going to be targeting with the guide RNAs for this experiment. This gene encodes for the A5 nicotininergic acetylcholinergic receptor subunit, and mutations there increase the risk for smoking and schizophrenia. Prefrontal cortex spontaneous default activity is altered in those psychiatric disorders, including schizophrenia. And the thought here is that perhaps nicotine and self-administration of nicotine via heavy smoking is a way to kind of self-medicate by some of these patients who are experience schizophrenic symptoms. So here we take a look at some of the data indicative of hyperfrontality in mice. VIP expressing neurons typically have a certain level of spontaneous activity. And then we look at VIP expressing neurons that have been applied with a guide RNA targeting chRNA5, which is the gene implicated in schizophrenia. We see a reduction in the activity of those VIP expressing neurons. That's the data that you see on the left. And it's really represented as a decreased level of spontaneous calcium transients. The data you see in figure E is a different representation of the same data. It's population averaged, indicating, again, a reduction in spontaneous firing rates. And figure F here, the median firing rate is reduced in chRNA5 knockout mice. So these are actually not chRNA5 knockout mice. These are mice which have had lentivirus injected into layer 2, 3 of the prefrontal cortex. And then when we look at a test of their hypothesis here, which is that perhaps nicotine would impact this and relieve some of those symptoms of hypofrontality, we see that cells in mice that have the alpha-5 SNP modification of chRNA5 or a deficiency there have a decrease in spontaneous activity of the VIP expressing neurons. We see that decrease can be rescued through administration of nicotine. That's the data you see there on the left, and the data on the right here is a kind of a time course across C, D, and E, looking at the reversal of hypofrontality, and again, the increase in spontaneous activity of those neurons through administration of nicotine over 2, 7, and 14 days. So there are a number of different formats that our CRISPR products are available in, plasmids being the most common format, but again, also in vitro transcribed RNA, which is suitable for in vivo microinjection, and the lentivirus, like I just talked about, which is good, can be used for screening in difficult to transfect cells, as well as in vivo injection. I wanted to take a brief moment to talk about a few new products that we have coming out that we're really excited about. These are in protein format, so these are delivered into cells and have activity immediately. Can be used for microinjection or transfection. And they're available as wild-type Cas9, Cas9 nickase, and enhanced specificity Cas9. The two products I want to talk about today are two that have come out very recently. The first one being FN Cas9, which is a different species of Cas9 from Francisella novicida. One of the key characteristics of this version of Cas9 is that it creates a sticky cut at the side of the double-stranded break, as opposed to wild-type Cas9, which creates a blunt-ended cut there. And we also have come out with a new dCas9 protein, which is fused with 3X flag peptide as well as biotin. These have a couple of different applications, so I'll show you some of the data that we have coming out with these two proteins that have been out in the last couple months. Francisella novicida Cas9 we found to be particularly useful for direct cloning of large genomic fragments, and largely because of that sticky end that's generated after the double-stranded break in DNA. It doesn't have the same high level of activity that wild-type Cas9 does on cells, but it has actually a higher activity in vitro applications, where it's just simply digesting DNA and performing molecular cloning experiments. Here we have a look at some of the data here, showing FN Cas9 RNP promoting more effective cloning of a large 12 kb LAC-Z locus as compared to wild-type SP Cas9. Cloning here performed in the E. coli bacterial strain BL21DE3. Next, some data on our dCas9 3X flag protein. This is particularly useful in chip assays and chip pull-down experiments. Here we will take a look at a few different markers at the P21 locus. We're looking at a pool of 3 different guide RNAs, bringing in Cas9 to target near there. And so when we use different antibodies, flag antibody, biotin, Cas9, as well as a p53 antibody, and DIG, which was actually what we use to label the tracer RNA, we see that these chips show specific enrichment of all of those different antibodies on the p21 promoter, just like p53, over non-specific antibodies. And fold enrichment here is calculated as specific antibody over mouse IgG non-specific. And then another representation of the same data where we're looking at percent input. The chips, again, here show specific enrichment of flag, biotin, Cas9, and DIG on the p21 promoters, similar to p53. p53 binds p21, and we can see that Cas9 and the elements that we've attached to that Cas9 protein are binding there very specifically as p53. The percent input here demonstrates the yield of chip DNA attained by a specific antibody. So with that, I'll be happy to take any questions. That concludes the presentation.
Thank you, Jeremy, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on the screen and click the Send button. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, how much lentivirus should be used in an in vivo injection?
So this question probably references that last publication that we showed. It's difficult to say exactly how much. When you're using lentivirus, you always want to determine a functional titer, because we know that the titer that's determined from these ELISA assays is not always representative of the functional titer. But in that experiment, they used 49 nanograms of p24 protein in 1 microliter locally injected into the brains of mice.
Jeremy, how many off-target sites should be considered for analysis?
So that's a great question. You can look at any number of off-target sites, really, because the methods that we use to predict off-target sites are not entirely comprehensive. But if we do use the methods that we have available to us, at a minimum, you want to take a look at the top 10 predicted off-target sites for the guide RNA that you're studying and that you're performing your experiments with.
Our next question is, how long should I wait to assess the phenotype from the gene knockout experiment?
Okay, that really depends on the format of CRISPR that you're using. So plasmids will require transcription and translation in order to have an effect in cell. Sometimes that can take as little as 6 hours or as much as 24 hours. Lentivirus, again, requires the same thing. Our RNP products, our ribonucleic protein products, are active immediately upon delivery. So those the effects and genotyping can be done in as little as 6 hours after the use of those plasmids. So dependent upon the format of the reagent you're using, much sooner when using an RNP than when using a plasmid or a lentivirus.
And Jeremy, it looks like we have time for one more question. What is the preferred method of delivery for RNPs in neural stem cell lines?
I think they can be delivered and with lipid-based delivery technologies that you might be familiar with, but there's not quite as efficient as electroporation-based delivery technologies. We prefer to use nucleofection here at Sigma, but really any type of electroporation-based technology would be best for the delivery of ribonucleic proteins or RNPs.
I would like to once again thank Jeremy for his presentation. I would also like to thank MilliporeSigma for making today's educational webcast possible. Before we go, I'd like to let everyone know that today's webcast will be available for on-demand replay through June of 2018. You will receive an email from LabRoots letting you know when your webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.